Hello, everyone. A very warm welcome from Garching, from Germany. Good afternoon or good morning to those of you joining from Europe, also EMA region and other parts of the world. I am Laila Elhash, and I'm a marketing manager at MSAID, and it's a privilege to have you all with us for our first webinar. Please be advised that this webinar will be recorded for documentation purposes. So today we have an exciting agenda lined up for you, and we're glad to share that our CEO and our COO will be guiding us through the whole seminar. Dr. Martin Frejno, our CEO, earned his Doctor of Philosophy in Oncology at the University of Oxford in collaboration with Professor Bernhard Costa at the Technical University of Munich, where he expanded his research to include bioinformatics and also large-scale characterization of cell lines phosphoproteomes. He will be guiding us today through the whole information regarding CHIMERYS 2.0 and share the astounding features of this latest release, and also will answer all your questions related to CHIMERYS. Following to that, Dr. Daniel Zolg, our COO, received his doctorate of natural sciences from the Technical University of Munich. During his studies, he led the generation of largest resources of synthetic standards called proteome tools. His work focused on exploiting the unique resource of creation of molecular and digital tools for proteome research. So Daniel will provide us today with a live demo session. He will also walk us through a live technical demonstration and also practical applications set up in CHIMERYS. And at the end of the session, he will also answer all your questions. Then we will wrap up the seminar. And I guess also from my side, thank you for joining us today. We also encourage you, all of you, to ask, or there is a Q&A sessions here in the chat. You're more than welcome to just post your questions there through the whole webinar. And with that being said, I will now hand the virtual stage over to Martin. I will just stop sharing my screen so that he can take center stage and also share his presentation with us.
All right, thank you very much, Laila. I'm now sharing my presentation. I hope everybody can see that. Please give me a thumbs up, Laila, if you can see the slide, the initial slide. Perfect. All right, then it's my pleasure to welcome you all as well in the name of everybody at MSAID for our webinar where we are going to introduce CHIMERYS 2.0 to you. But not only CHIMERYS 2.0, I will walk through all the various different pieces of software that we at MSAID develop and integrate into other software packages, and tell you a bit about what they are, what you can use them for, and how they might be of benefit for your particular question of research. Just a bit of trivia. Frequently, people pronounce MSAID as MSAT. That's, of course, completely wrong. It's MSAID for a reason, and the reason is we are aiding mass spectrometry with AI, and it's this topic that spans the entire development process of our software tools, and the goal is to basically revamp heuristics that we've been using for the past 25 years during the analysis of bottom-up proteomics data in order to allow you, the researcher, to extract as much information out of your raw data as possible. So without further ado, I will jump straight into our suite of software tools, and these are three of a kind, and all of these three software tools are integrated into Proteome Discoverer. So it is very seamless for you to access them through the interface that you are used to. And these three software tools are Inferis Rescoring, and CHIMERYS. I will basically throughout this presentation introduce these three tools one after the other. I will share its unique capabilities and what we recommend that you use it for. And at the end, of course, there's time for questions of all sorts, and we are happy to answer them as much as we can. All right, so let's dive straight into the first topic, and that is inferis. Inferis is a deep learning tool for the prediction of mass spectra. But why would we be thinking about deep learning tools in the first place? In order to answer that question, one slide that I typically always show is this slide here, and it is basically here to visualize that in mass spectrometry data, as you know, we record two-dimensional data. We record the presence and absence of fragment ions on the MZ axis, and we record their relative intensity on the Y axis. But traditionally, the majority of all database search engines have been analyzing actually only one of these two dimensions, and that is essentially the MZ dimension. So typically, when you have an experimental spectrum like this with B and Y ions, what a typical search engine would do, it would calculate for a given peptide candidate that would potentially be able to explain this pattern of fragment ions. It would calculate all possible theoretical fragment ions, and it would then score the presence and absence of every one of them, and then do a tally of how many fragment ions have we matched, how many fragment ions have we not matched, and then calculate a probability-based score that basically answers the question, how likely is it that this particular signal that we see in the MS2 spectrum can be explained by this particular peptide. But as I mentioned, we record data not in one dimension, we record it in two dimensions. So naturally, wouldn't it be great to actually access that second dimension in order to identify peptides as well? And that dimension is, of course, the intensity dimension. And as you all know, if you record the same spectra for the same peptide on the same mass spectrometer over and over again, the characteristic pattern of fragment ion intensities doesn't really change. So there's a characteristic fingerprint each peptide will basically produce in a mass spectrometer. And it is this intensity-based fingerprint that we would like to make use of in order to identify peptides. Traditionally, that has been made accessible through the use of spectral libraries. And what these let you do is basically calculate a similarity of a library spectrum, so a spectrum that you've previously acquired and annotated with a peptide identification in some manner, to a new experimental spectrum. And you can then calculate intensity-based figures of merit. And these intensity-based figures of merit really allow you to increase your sensitivity when it comes to the identification of peptides in MS2 spectra. However, spectral libraries have, of course, one essential drawback, and that drawback is that you have to generate such a spectral library. And for those of you that are using DIA as an acquisition method, you will know that the best library is always the library that you've acquired on the data that you want to analyze in DIA. So always doing the measurement of the same sample twice is very expensive, very time-consuming. So researchers have been trying in the past to sort of get around that library generation step with various different ways of library-free based data analysis. But at the core of the most recent innovations is that you can now actually use deep learning models to predict fragment ion intensities rather than acquire spectral libraries that you then use for data analysis. And the main driver behind this are projects such as the Proteome Tools project, which generate vast amounts of high quality training data for neural networks to essentially learn what it means for a peptide to be fragmented in the gas phase. So how does that work? Every deep learning model is just as good as the data that you use to that you train it on. So you have to start with high quality data. We here at MSAID, we train our models on Proteome Tools data and other data from the public domain. We use PRIDE, for example, as a source of high quality spectra. And then what we do is we perform data selection and curation. So we only select very good identifications, very high signal-to-noise spectra in order in order to incorporate them into our training data set. And then we run what we call an AutoML pipeline, where we basically take all that training data, we train thousands of different models with different configurations, and then select from these thousands of models the best performing models and convert them into an appropriate representation for you to be able to use them later on in applications such as, for example, Proteome Discoverer. So what can you expect from such a tool? Such a tool and our tool that our model is called Inferis. What is Inferis capable of doing? That is visualized here in an example on the right-hand side, where we have an experimental MS2 spectrum on the top with its annotated B and Y ions, and a predicted spectrum at the bottom. And as you can see, the prediction performed by Inferis is virtually indistinguishable to the experimental spectrum. And that we measure this similarity based on, for example, the Pearson correlation. And this here is a Pearson correlation of 0.99. So the two spectra are virtually indistinguishable of one another, which means we can use these fragment ion intensities as priors for identification in different tools when it comes to assigning a peptide sequence to an experimental spectrum. Inferis has been part of Proteome Discoverer since 2020. We started with our Inferis 1.0 model in 2020. And since then, we've basically increased our capabilities with Inferis 2.0 in 2022 by incorporating charge 1 precursors. So we increased our performance in that realm. And by now, this year, we've released Inferis 3.0, which is integrated into Proteome Discoverer 3.1. And this model can basically predict peptides that were generated by any protease between 7 and 30 amino acids in length. We support charge states 1 to 6, CID and HCD fragmentation. And by now, and I'm sure many of you will like that, we support phosphorylation as a dynamic modification, as well as oxidation and carbamino methylation. And you can analyze TMT and TMT Pro labeled peptides, as well as SciLake labeled peptides. Inferis, as incorporated into Proteome Discoverer, can be used to predict spectral libraries. And those spectral libraries you can then essentially use in tools such as MS-PepSearch and PD in order to analyze your data that way. I would like to highlight one key innovation that we've introduced now with Inferis 3.0, and that is essentially the capability to predict phosphorylated peptides. We didn't only include more training data here, but we also decided to include additional ions apart from only B and Y ions that when we predict these spectra, because they explain a substantial amount of experimental intensity in our experiments. The key innovation here is basically that we, in addition to B and Y ions, we have now the loss of ammonia, we have water losses that we predict, and also phosphor losses. And by virtue of including these neutral losses as predictions, we can now explain as much as 50% more experimental intensity with the predictions that Inferis performs in comparison to earlier inceptions of our deep learning model. So now you might ask yourselves, what can I use these predictions for? Yes, I can run MS-PepSearch, but can I do anything else with them? And the answer, of course, is yes. And one key application that I would like to highlight here is Inferis rescoring. Inferis rescoring is essentially a rescoring workflow that is end-to-end embedded into Proteome Discoverer. Just the way that you were using sequenced HT before, you can now use Inferis rescoring in addition in order to boost the identification performance of sequenced HT. The workflow that you would be using for that is depicted here on the left. And the only difference from your classical sequenced HT workflow is that with Inferis rescoring, you slide in the Inferis rescoring node in between sequenced HT and percolator. So the general idea of this workflow is that we come in with a set of experimental MS2 spectra from a raw data set. we allow Sequence HT to score these spectra in an intensity-free fashion. And then not only the top hit, but actually the top up to 10 hits are reported to Inferis rescoring. And for these hits, Inferis predicts the fragmentation spectra and then calculates intensity-based figures of merit. And these intensity-based figures of merit are then used to basically increase the sensitivity in separating true identifications from false identifications. One key benefit of these intensity-based figures of merit, such as the Pearson correlation, but also the spectral angle, is that they are much more stable over the elution curve of a peptide. This is visualized here on the right-hand side, where we have an XIC of a typical peptide. The area here is shaded in blue, and we have the relative intensity on the y-axis. And when you plot, in addition, the relative so cross correlation score, the relative number of matched fragments, or the relative binomial score, you see that these are very sensitive to the amount of peptide that eludes off the column at a given point in time. And these are all intensity-free figures of merit. But the spectral angle, which actually makes use of the predictions from Inferis, this is now a lot more stable over the retention time, which really allows you to dig into the weeds of your data set and pull out identifications that are very low in intensity. So what do we recommend Inferis rescoring for? Well, one prime use case for Inferis rescoring is to boost the performance of Sequence HT on immunopeptidomics data. And here I've brought you an HLA class 1 data set, which we've analyzed with Sequence HT. In Proteome Discoverer, or with Inferis 1.0 or Inferis 2.0 rescoring. And as you can see, when you consider the number of identified PSMs here on the left-hand side, or the number of identified peptides on the right-hand side, that we substantially increase the amount of identifications when you use Inferis rescoring in addition to Sequence HT. The bump from Inferis 1.0 to Inferis 2.0 basically comes from the fact that now with Inferis 2.0 and 3.0 by extension as well, we also support a much more improved performance on charge 1 analytes. But of course, you can ask the question, well, these additional identification, this is a substantial increase from Sequence HT of 68% in the number of identified peptides. Are these actually true identifications? And the answer to that is yes, they are. And the way we judge this basically is by looking at the sequence motifs of the identified peptides. This particular data set here was taken from a study from 2020, where the authors have worked with monoallelic cell lines that basically should only harbor a particular HLA allele, and that HLA allele will only result in a particular set of HLA peptides to be identifiable. When we separate these identifications now into identifications that we were able to obtain using the classical workflow, so just Sequence HT, or with rescoring, these two sequence motifs are virtually identical. And if you look at the rescoring exclusive identifications, so the IDs that you were only able to get because we were using Inferis rescoring, these also reflect the expected sequence motif, which strongly suggests that these are true identifications. An additional use case for Inferis is also to boost the identification performance on single-cell Eintrap-based data. So here we have a data set that basically is a raw file of a single motor neuron. And here, what you can clearly see is that with Inferis rescoring, we substantially increase the sensitivity in this single cell data set. And it is also very evident why that is. If you look here at the example on the right-hand side, this is a typical spectrum. On top is the experimental spectrum of a peptide identification that, when you use sequest, was not able to make the FDR cutoff of 1%. And the reason is clear. I mean, there's the fragment ions are very low abundant, and there's a whole bunch of other fragment ions that are present. the cross-correlation score alone wasn't sufficient to promote this identification below the Q-value threshold. But if you now use Inferius rescoring and include the spectral angle, so an intensity-based figure of merit in addition to the cross-correlation score as a feature in your FDR estimation, what you can clearly see is that now this identification is below the Q-value threshold, meaning this is now an identification that you were only able to make in that single motor neuron because you've used that second dimension in your mass spectra with the help of Inferius rescoring. All right, so now I've told you about two tools that have been in Proteome Discoverer since version 2.5, since 2020. And now we come to the elephant in the room, which is CHIMERYS. And I would like to go into why you would care about CHIMERYS and how CHIMERYS actually makes most efficient use of the predictions that Inferius rescoring supplies. So at the inception of MSAID, we were basically faced with this particular problem here. It's not a problem that is unique to us at MSAID. It's unique to everyone that measures mass spectrometry-based bottom-up proteomics data, and that is that chimeric spectra are very prevalent even in DDA data. What is a chimeric spectrum? Basically, that's a spectrum that contains fragment ions of multiple precursors. And in order to visualize this problem, we usually use a plot like this, where we look at a spectral library of roughly 400,000 precursors, and we just plot the number of precursors in bins of 2 Thomson by 1 minute, and just count how many precursors we have in these bins. And as you can see, the majority of the entire gradient that you would measure here on the y-axis and the majority of your MZ space, you would basically, wherever you place an isolation window here, you would be isolating multiple precursors and would be generating chimeric spectra. So in DDA, this is somewhat of a nuisance, but when we go to wide window acquisition, depicted here, or even to DIA, we intentionally generate chimeric spectra, and hence we have to solve them somehow. And that problem is, of course, not new. It's a relatively old problem and has been tackled in the literature in various different ways. Here we depict 2 of these ways. We call it the subtraction and the duplication approach. And both of these ways are actually a bad approach to the problem of chimeric spectra, since they either use information of fragment ions too often, or they use it not often enough. In essence, the subtraction approach will identify a single PSM, remove the identified peaks from the experimental spectrum, and then submit the remaining peaks to the search engine again with a different precursor mass. And the duplication approach will duplicate the full spectrum for each precursor mass in the isolation window, and then separate these, then submit these copies of spectra separately from one another. This is, by the way, the approach that is implemented in Proteome Discoverer with the precursor, with the MPS node. Right. So when we were faced with that problem and saw that traditional approaches sort of had drawbacks in the way they approached that data, we came up with a completely new way to deconvolute chimeric spectra. And we do that with the help of our predictions. And the entire philosophy behind this is that we want to explain as much experimentally observed intensity in a particular MS2 spectrum with as few peptides as possible. And when we do that for an experimental spectrum like this here, for example, we can not only identify 1 peptide, but we can identify 2, 3, and even 4 peptides in this particular experimental spectrum. And you can for sure appreciate that the fragment ion intensities, even the ones of shared fragment ions, such as the Y1 here, they align very well. The predicted ones align very well with the experimental intensities here at the bottom. And if we now ask the question, are these peptides even remotely similar to one another? We can predict their retention times, and we can see that they're predicted to elute roughly at the same point in time. And if we look then in the MS1 isolation window that was used to acquire this particular spectrum, we see that all these 4 precursors are present. and were indeed co-isolated and co-fragmented to generate this particular experimental spectrum. So we basically use linear algebra to deconvolute this experimental spectrum. And in the process, we can calculate contributions of each individual peptide to the total ion current of that experimental spectrum. And this is then later on the basis for our quantification. And I will go into a bit of detail there later on. So how does CHIMERYS utilize these new capabilities? Well, CHIMERYS is separated into 4 steps. We have an intensity-based scoring using our predictions, where we generate a couple 10,000 PSMs that we then use for the recalibration of our deep learning models. Then we have our deconvolution step, and then afterwards we have error estimation, so FDR estimation using Mokapod. And with CHIMERYS 2.0, that is now part of PD 3.1, you can analyze any data from any acquisition method. So out of the box, CHIMERYS 2.0 can be used to analyze DDA, DIA, and PRM data of peptides generated with any protease that can be label-free, TMT or TMT Pro labeled, or SILAC peptides, and we support phosphorylated peptides. So what advantage do you have when using CHIMERYS as opposed to other search engines? Well, given the fact that CHIMERYS is very good at deconvoluting chimeric spectra, we can substantially boost the number of identifications in typical DDA experiments here in this case. So here we took a 1-hour DDA HeLa lysate measured on an Orbitrap Explorers 480, and it was acquired with 1.3 Thomson isolation windows, and we analyzed that data with sequus-HT, sequus-HT with precursor detector. This is the duplication approach method that I mentioned earlier, implemented in PD. We use Inferis rescoring here in light blue and then CHIMERYS in orange. And you can immediately appreciate that the number of identifications that you gain when using CHIMERYS is substantial. We are basically doubling the number of peptide identifications in this particular case, and we add 50% more protein identifications as well. But if you're not after the greatest depth of your particular study, you can also decrease. You can also increase your throughput by decreasing your gradient length. This is visualized here on the right-hand side. We compare now sequus-HT against CHIMERYS in orange. And as you can see, you can basically identify the same number of peptides in a 14-minute gradient that you were previously able to identify with sequest in a 46-minute gradient. So that is a nearly a factor 3 of throughput increase on the back of simply a different way of analyzing your experimental data. Of course, we not only compared CHIMERYS to sequest, we also compared CHIMERYS to other types of softwares. And here on the left-hand side, I brought you a comparison of CHIMERYS versus a whole host of different search engines that are typically used to analyze DDA data. Here we are working with a different public data set that was published in 2022. And you can see that CHIMERYS basically has nearly 25,000 unique peptide identifications that no other of these search engines were able to identify. And it shares basically, it basically encapsulates all identifications, nearly all identifications that you are able to generate with these other search engines. And we do that basically on the back of these intensity-based figures of merit. So the key takeaway is CHIMERYS can confidently call a peptide identification even on fewer fragment ions because we have that additional dimension of our MS2 spectra that we can analyze the intensity dimension. So you can see that the peptides that are unique to CHIMERYS basically have fewer number of peaks, but they maintain a very high spectral angle. So we can basically rescue them by the virtue that their expected fragment ion intensity distribution is reflected in the experimental MS2 spectrum. So this was already available in PD 3.0. So DDA data you were already able to process with PD 3.0, but now with PD 3.1 and the capability to analyze phosphorylated peptides, you can now use CHIMERYS also to analyze your phospho data sets, your phosphoproteomes. And when you do that, what you gain is basically a substantial amount of identified phosphopeptides. And this is visualized here in this particular plot. The workflow is depicted here on the left. Basically, we start with the spectrum files, just have the spectrum selector in between, and then CHIMERYS. And when you do that, you basically increase the number of serine containing peptides, threonine containing peptides, and tyrosine containing peptides in this particular data set. And the overlap between CHIMERYS and other search engines such as Comet and Sequus is substantial. So the key takeaway here is even when you're doing phosphoproteome experiments, CHIMERYS will basically allow you to increase the number of identified PSMs and the number of identified phosphorylation sites. What is very nice about the Proteome Discoverer environment is that now you can also basically chain CHIMERYS to the PTMRS node that gives you the localization of phosphosites. That works straight out of the box. So on your typical DDA experiment, CHIMERYS now is basically a drop-in replacement for Sequus when you want to analyze phosphopeptides. And when you use PTMRS, again, you get localization. And that localization basically then allows you to pinpoint where the phosphorylation site is, and even localized, so confidently localized sites, you have a substantial increase when you use CHIMERYS as opposed to Sequus HT or Comet. So now one brief slide on which I think is the best feature that comes with CHIMERYS, and that is basically that now CHIMERYS can analyze data from any acquisition method. I mentioned earlier that when we deconvolute our chimeric spectra, we calculate contributions to the total ion current for each peptide. And these contributions we actually use in the new fragment ions quantifier node in order to basically quantify peptides in DIA experiments based on their MS2 signals. So we have our identification here on the left based on a chimeric spectrum, and then we do that. What we do is we trace the contribution of the orange and the blue peptide over retention time. So each vertical line here is an MS2 spectrum. We calculate contributions for these peptides for each of these spectra, trace these contributions over retention time, and that gives us our quantitative readout for a given peptide in MS2. We allow for gaps in identifications as visualized here on the right-hand side. So even if you've missed an identification at a particular spot, we will still allow you to quantify, for example, here the tail of the chromatographic peak. And the really cool thing is that that CHIMERYS actually performs FDR-based peak picking. So you basically focus your quantitative readout in the area where we have FDR-controlled PSMs. And that is a unique feature that is only available in CHIMERYS when you analyze MS2 quant data with CHIMERYS. So how does that novel way of quantifying peptides in MS2 perform in comparison to traditional methods working on fragment ion XICs? Well, suffice it to say, it performs very well. Here on the right-hand side, we basically have the correlation of our coefficient-based quantification to the skyline-based quantification of 52 peptides in a PRM experiment. So this is now a PRM data analysis. And you see that we are basically, we basically have a correlation coefficient of 0.99, I believe. And we can even perform FDR control on PRM experiments on the back of co-isolated peptides. So in this particular data set, we targeted 52 peptides for analysis, but we've actually and we've actually identified more than 2,000 peptides that are co-isolated at the same time with these 52 targeted peptides. And here's an example for that. Basically, we targeted the orange peptide, has a beautiful elution curve, but we've also identified the blue, the green, and the purple peptide, which we could use to perform FDR control on that particular data set. So given that we can analyze PRM data in that way, you might ask yourself, well, a PRM scan is nothing else than a systematic measurement of a DDA window, DDA isolation window over time. And if you look at different spectra that are acquired at different isolation window widths, you can see that these spectra are fairly similar to one another. So a DDA spectrum with 0.4 Dalton MZ isolation window can be very complex, even as complex as a DAA spectrum that was acquired with 8 MZ isolation window. So naturally, we thought, can we extend the way we analyze PRM data in CHIMERYS also to DAA data? And the answer is yes, we can. So basically, if you need to ask yourself the question, do I acquire my data in DDA or DIA? That now becomes an optimization parameter of your experiment. You can do either, and you can analyze both data with CHIMERYS in a very straightforward fashion, just the same way as you would be analyzing DDA data. You can now also analyze DIA data in Proteome Discoverer with CHIMERYS. So how well does it perform? When we basically analyze peptides when we do any experiment, usually what we do first is we ask the question, can we be sure about our error estimation? So we perform entrapment experiments, which are also called double decoy experiments, where we spike in peptides that we know shouldn't be in the sample into our target FASTA file and don't tell the search engine about it, and basically try to quantify later on how many of these peptides that shouldn't be there the search engine wrongly identified. So this gives us a measure of empirical FDR, which we can compare to our target decoy-based entrapment FDR. And the comparison is here plotted on the left-hand side, where we have FDR on the x-axis, entrapment FDR on the y-axis. And you can see that in an ideal world, where you would want your graph to be is as close to the diagonal as possible. And with CHIMERYS, we are basically very, very close to the diagonal, meaning that our FDR that we calculate based on the target decoy approach pretty much reflects the empirical FDR that we measure with our entrapment experiment. So when we analyze this two-organism mixture data set that produced this particular graph, also with Diane and Spectronaut, what we can see is that if we filter at FDR or entrapment FDR, we can basically measure what is the decrease in identifications, number of identified precursors in this case, when we filter at the actual FDR. So CHIMERYS doesn't lose many IDs, and this is because FDR and entrapment FDR are virtually identical to one another. And the same holds true for Spectronaut. However, for Diane in this particular case here, we see that they basically underestimate their FDR, because if we filter the data set at 1% entrapment FDR, we lose a substantial amount of identifications that Diane claimed to have, bringing Diane basically on par with CHIMERYS and Spectronaut. So these three tools, when you control identifications at entrapment FDR level, perform essentially the same. So they perform the same on the identification side. How do they perform on the quantification side of things? Well, here, since we have a three-organism mixture experiment, essentially, we can basically look at the expected ratio distributions. Again, we have CHIMERYS, Diane, and Spectronaut, with the expected ratios delineated as dashed horizontal lines. And I'm sure you can appreciate that the MA plot of CHIMERYS, so log2 mean intensity of the precursor against the fold change, looks very similar for CHIMERYS as for Spectronaut. And that is also reflected in the ratio density plots here on the right-hand side, meaning that with this completely novel way of quantifying peptides in MS2, with CHIMERYS, we can essentially get the same accuracy and precision in terms of quant as Spectronaut gets. But the biggest advantage of using chimeras for this type of data is that you can now acquire the same sample in DDA and DIA and essentially analyze it with the same search engine, and that wasn't possible before. So now we again compare a sample that was acquired either in DDA or in DIA, analyzed with chimeras. And you can see that basically in terms of the number of identified PSMs, DIA performs much better on this particular data set than DDA. But in terms of the number of identified peptides, DDA outperforms DIA. And this is primarily due likely to the fact that this data set here is was acquired with relatively wide isolation windows. So we would be very curious how data like this looks on sort of more recent mass spectrometers, such as the Orbitrap Astral, where we can measure DDA and DIA with the same isolation windows, essentially. So then, how does the quantification in the DIA sample based on MS2 compare to MS1 based quantification using MANORA feature detector? The answer is very well. So again, these are two different raw files, one measured in DDA, one measured in DIA, and you see basically that the quantification in MS2 corresponds very nicely to the quantification in MS1. So basically, with that said, essentially what you can do is with chimeras, you can now analyze DDA, DIA, and PRM data of phosphorylated peptides, unlabeled peptides, and you can quantify as well in MS1 as you can quantify in MS2. So the key takeaways here are that DDA, DIA, and PRM data for the first time can now be analyzed with one software package, and that is CHIMERYS 2.0. And that is on the back of our deep learning-based predictions, which enable us to analyze that data in a spectrum-centric fashion. We have a completely novel way of quantifying peptides in MS2 using these contributions that I mentioned earlier, and that approach is essentially the same in terms of quantitative accuracy and precision as Skyline. Chimeras can perform automatic peak boundary detection based on FDR, which essentially abolishes manual intervention in your PRM data analysis. And for the first time, you can now do head-to-head comparisons of DDA versus DIA with the same software package. So all in all, I think we can predict a bright future for ML and proteomics. And with that, I'd like to acknowledge the team. I took way more time than expected. I'm sorry for that. So there's not as much time for questions. Hopefully Daniel was able to answer some of your questions on the fly already. But thanks for the great team. I'm just the one presenting. There's a whole host of people that actually make this happen. I would also like to thank the team over at Thermo Fisher, the Proteome Discoverer software team. And of course, if you want to try chimeras for yourself, you can request a free demo to see how it works on your particular data set. Thank you very much.
Thank you, Martin, for your presentation. Well, I see two questions that haven't been answered, so I will begin by addressing the questions. The first, any plans to include ETD, ETHCD predictions as well?
So that has been on our radar. ETD and ETHCD is something that is not a very high priority at the moment on our end. ETD predominantly is being used for the analysis of phosphopeptides. In our hands, HCD performs also very well there. So for the moment, I would say this is a bit of a ways out, I think.
Thank you very much. And the second question, how do you deconvolute the contributions of each chimera in the spectrum to the label abundance in TMT data?
So in TMT data, we don't perform deconvolution of the TMT intensities. What we assume when we deconvolute the chimeric spectrum is that a chimeric spectrum is a linear combination of pure spectra. So that is the basis of calculating the contributions that I mentioned earlier. And what these contributions allow you to do, in particular for TMT data, is that you can filter your PSMs to only those PSMs where the majority of the contribution can be attributed to a single peptide. So essentially, you can use CHIMERYS data as sort of a precursor impurity filter, which is not calculated on MS1, but is calculated actually on MS2. And that is a big advantage because traditional methods that assess precursor impurity and ratio compression based on this rely on a signal in MS1, but you can still get ratio compression if the precursor is not measurable in MS1 anymore due to dynamic range in MS1. So yeah, this is a key feature that you can use in TMT MS2 based workflows with CHIMERYS.
Thank you very much, Martin. So I have another one. Any plan to include other modification like acetylation or methylation?
Yes, it's on the horizon.
Awesome. So I guess I will continue now with the second part of this webinar. So I will now hand over to Daniel, who will provide today with a live demo session.
All right. Also, welcome from my side. So I see there are a couple of questions that have just popped up in the chat. I believe Martin will try to answer a couple of these so they're not lost. I would like to take a couple of minutes to walk you through how to submit a CHIMERYS 2.0 search in Proteome Discoverer 3.1. I will quickly talk about the setup and the login into the CHIMERYS service. I will then start a search and we will have a brief look at the results. So here I have a Proteome Discoverer 3.1 installation on a VirtualBox in this case, where that I got from the Thermo demo download portal. If you need links to that, or if you have not yet registered for a CHIMERYS demo, you can just do that through our website or a website on the Thermo Fisher platform. We will provide links to both of these pages in the chat. So first of all, after you installed your Proteome Discoverer demo version, you will note that you have to log into the service to utilize CHIMERYS. You will have received a login credential in an email that we will send you after you requested your demo credentials from us. These demo credentials are good for approximately 24 hours of processing, and if you need more time, obviously we can help you out with that if you need a bit more files to figure out if CHIMERYS is actually helpful for your research. So what we have to do is, before we go somewhere into the setting up the workflow, we need to make sure that we're logged into the CHIMERYS service. And for that, you have to go to Administration tab and go to the CHIMERYS tab. And you will see that here in this case, there's a green box. So I'm logged into the service. If I'm not logged into the service, I can go on these three dots and put my demo credentials that I received in my email. After that, I'll just have to click apply. In this case, I'm already logged in, so this doesn't turn green here anymore. After that, we're ready to start our first analysis, and I would like to walk you through the parameters of the search and walk you through a couple of hints and tips and tricks that we've encountered when using CHIMERYS. So let's create a new study here. I'm just gonna call it study 2. I'm gonna put it onto my desktop, and I will quickly add a couple of small DIA raw files that I've prepared. Please note that these are relatively small replicates of DIA, just 5-minute runs, and you will realize that they're just 5-minute runs because when we look at the quantification later, you will see that you don't have a lot of data points across the peak. We might want to acquire your data in a way that you have a little more confidence in your quantification due to more data points. So we can just create a new analysis, and we're greeted with the usual window and first configure our processing step. So what's happening is that you can either use any of the common workflows that are already provided. If you haven't seen it, there are a couple of workflows already pre-made with CHIMERYS and respective settings. In this case, for example, TMT Pro and CHIMERYS, but I want to do this manually from the beginning. So first of all, we're just gonna need our spectrum files node. And in general, we do not recommend using the spectrum file recalibration node that some of you people might know, because for CHIMERYS alone, it does not matter if you recalibrate your spectra beforehand, because we perform data-driven recalibration during the process. The only reason why you might be wanting to use the recalibration node is if you suspect that your raw files are heavily miscalibrated and you're trying to use MS1 quantification using the minor feature detector. In this case, recalibrating these features might help you get better quantitative values. But just for identifications, we don't care about any recalibration that is performed. And this is basically compute that is extra and does not need to be performed in most of the cases. The next one would be then the spectrum selector, where we don't need to change any of the settings, and in the end, CHIMERYS, which I'm going to connect to my spectrum selector. You will already note that in contrast to CHIMERYS 1 in Proteome Discoverer 3.0, we do not need to connect a percolator node here. This is not necessary anymore because by now all the FDR estimation is performed in the cloud, and we have switched over from a tool called Percolator to a tool called Mockapod, which is a reimplementation of the same principles, which was spearheaded by Will Fondry. And we have edited in a way that it's much faster than the initial percolator that was used in CHIMERYS 1.0 or PD 3.0. So if we now go through all of the configuration settings of CHIMERYS, I'll tell you a little bit about how to set this up. So first of all, you have a couple of general questions that you're asked in your workflow. And the first one is, do you want to process your files all together? And that also means that you would use Mockapod on all of your results together, or if you want to spawn them job per file. Usually we recommend just to have one job for all files. The cloud service will automatically take care of the parallelization for you, meaning that if you submit multiple jobs inside the same, multiple files inside the same job, we will automatically parallelize over the respective raw files to speed up the analysis time. There's also an option called failed job case, which you don't need to necessarily change. This basically handles what happens if one of the pods is not able to completely process the file, which is usually only the case if you have, for example, very strong fractionations where your edge fractions in your 2D dimensional fractionation is basically empty and cannot completely be processed by CHIMERYS, because we needed a couple of data points to recalibrate our models. And the next one is just if you want to store your fragmentation traces and if you want to store your quantification traces, and you will see that this is quite useful when we look at the results. The next one would be to provide an input database. In this case, I'll just have pre-configured some SwissProt human FASTA, and you can select a prediction model. Note that there are two prediction models available in CHIMERYS 3.1, in CHIMERYS 3.0, in PD 3.1. The first one is in Frejno 2.11, which you already know if you have had Proteome Discoverer 3.0 and CHIMERYS 1. And the new one is the Frejno 3.1 fragmentation model, which features fragmentation of phosphorylated peptides. So for people who still want to use the old model that also uses a different set of fragment ions, because we did not predict neutral losses, losses of phosphorylated side chain residues, water loss, and loss of ammonia, you can use the 2.11 model. In this case, you cannot select phosphorylation as a modification. If you want to have the benefit of using the new model, you just select it, and you can then select in your dynamic modification settings also, for example, phosphorylation on S, T, and Y. The next one is a field which is called CHIMERYS inclusion file. I want to jump this for now, and I want to talk about this a little later once we have submitted the first search. Obviously, then you have your parameters that are well known from other search engines. You can configure your enzyme, your miscleavages, your min and max peptide charge. If you want to save a bit of runtime, you might also want to turn down your maximum peptide charts from 6 to 4, because usually in tryptic digests, we do not encounter a lot of highly charged peptides. You can select your fragmentation mass tolerance, either in ppm or in Dalton world. If you switch over to Dalton and if you're analyzing ITMS data, we usually recommend using a tolerance of about 0.3 Dalton and not much bigger. Then you can configure your modifications. As you can see by now, the list has grown to quite some modifications. Obviously, we're also striving for adding more PTMs as new models come in, so we increase compatibility with your research question. You will see that you can also put in labels like TMT and TMT Pro, which can be added as static modifications. In the current version, carbamide methylation is still a required modification, but we're working heavily on getting rid of this limitation in one of the following releases of Inferis. And then in the end, you can change, but we do not recommend your target FDR for validation. So in this case, we've just now configured a very simple run. I'm just gonna add my raw files and then quickly move over to the consensus step. In the consensus step, I just want to show you what is important if you have quantification in your data and you want to get that out. So here in the basic workflow that you can select through the usual interfaces, you basically would then need to select one of the different ways to quantify your data. In the case that you have DIA data, the only thing you have to do is you just have to pull in your fragment ion quantifier, which will automatically create fragment ion traces based on the contribution for each peptide. And with that, we can already start our search. In case you do not have DIA data or have any other data like PRM or DDA, you do not need to change anything. You do not need to set anything. Everything will be automatically determined. And with that, we can basically start our search. The search will now complain a little bit that I did not configure study variables for the quantification, but we can just ignore this now, as these are just 3 replicates of the same HeLa sample. So before we exit this live demo, I just want to show you 2 things which I think are very nice in terms of looking at your data. And the very first thing is that if you now go into your results and for your convenience, I've already processed this results for you so we can look at them because they won't finish in time. You basically can, oops, let me just hide these again. You basically can go into your peptide groups. You can enable your chromatogram traces and you will see the fragment ion traces which are aggregated into this contribution quantification for all 3 replicates here. As you can see, this is a very short run. It's only 3 minutes and you don't have a lot of data points across the peaks, but you can see that there's a little retention time shift between your first and your second and the third replicate. And you can basically look at these for every single peptide that you identify. However, if you set up your methods, please, please, please make sure you have a little more than 3 data points across the peak. This is also something which is super important in DIA data, where you're basically trading IDs against your quantitative accuracy. So just optimizing for IDs in DIA is not a good idea. You always should look at your quantification traces if you want to get high quality data. And the second thing that is nice is that CHIMERYS is still very much a ESM-centric search engine, meaning that also in DIA data, you can look at individual peptides and how they fragment. And how you can do that is you just open that by clicking on any PSM that you want to see, and we will automatically generate this mirror plot for you. And while there's quite some stuff happening in the experimental spectrum, you can clearly see that the fragment ions for this one nicely align, and this is an identification you can trust. And this separates CHIMERYS from any of the other search engines which do not allow you to look at individual spectra to verify what you have actually detected in your sample. And you might want to do this before basing a postdoc or any of the follow-ups on identifications that you get in your DIA data. And with that, I'm already a bit over time. I would like to close this demonstration and obviously also take some questions. This is not a comprehensive Proteome Discoverer seminar. There is a Proteome Discoverer seminar coming up in Bremen in the beginning of December. If you're interested in that, this and a lot more will be showcased there, including much more in-depth discussions and tutorials on Proteome Discoverer itself. If you're interested in learning more about this, we can also schedule a follow-up seminar where we talk more in depth about Proteome Discoverer, how to set up studies, and how to view these studies.
Thanks, Daniel, for the live demo session. And also thank you, Martin, for helping me answer the questions, and you too, Daniel. Well, I don't see any more questions in the chat. Okay, just last one, just pop. In the demo, there is no consensus WF for select data. How to do this and retain also individual channel intensities?
That's a good question. It's hard to answer without showing you. What I would do is I would quickly configure a workflow for you and send that to you, or include that in the email after this seminar. So this is easily possible. You can basically would configure your search in a way that you have variable modifications for both your heavy on your heavy channel and for your light channel, and you would get for every peptide quantitative readout for both of them. And how am I exactly able to see data points across the peak? Well, I just looked at that, and back in my days, I still learned that you need 10 data points across the peak to have nice chromatographic peak shapes. Some of the peak shapes I just showed you were just basically triangles where you have 1 or 2 data points and then it goes back to 0. Usually what you want to have is that you have somewhere between 4, 7, maybe 10 data points across the peak. And whatever I say right now very likely will spark discussions with people who are adamant about QC in liquid chromatography. More is generally better.
Absolutely. Thank you very much, Daniel. So I also posted on the chat, like if you have any further questions, just reach out to us to our email address and we will be happy to assist you. So, and with that, I guess I hope you gain valuable insights about CHIMERYS and also how to install CHIMERYS. And yes, right now I'm just going to share my screen, and we invite you to request a free demo by scanning the QR code or just visit us at msaid.de. If you haven't tried CHIMERYS 2.0 yet, this is your opportunity. And thank you all for joining our webinar. Thank you, Daniel, and also thank you, Martin. And we will ensure the recording is available on our YouTube channel for your convenience. And stay tuned for more exciting updates from MSAID. Thank you and have a wonderful day. Bye.
Thank you, everyone.
Bye-bye.